Alright guys, so now we're going to go over the two main processes of the central dogma theory of protein synthesis. Now all of this that I'm about to describe is how it takes place in a eukaryotic cell, so cells that have membrane-bound organelles. It's very similar in prokaryotic cells, but there are a few differences. For example, prokaryotic cells don't have a nucleus, so their process doesn't really require anything to leave the nucleus because they don't have one. So they don't have quite as many processes as we do in eukaryotic cells, but we really are only going to talk about transcription and translation in terms of eukaryotic cells, because yeah. So just a quick overview of the difference between the two processes. Transcription is going to occur in the nucleus, where translation is going to occur in the cytoplasm. Transcription is whenever we are making that mRNA, and like I mentioned earlier, it's really similar to DNA replication, but you're only using one side of the ladder or one side of the DNA double helix. And you're going to use RNA polymerase instead of DNA polymerase to add those RNA nucleotides. And then in translation, the mRNA is going to leave the nucleus after transcription, and it's going to sit on the ribosomes with the nucleotides facing up in the ribosome. If the nucleotides were facing down, then they would be blocked and the anticodon of the tRNA would never be able to find them. So they're going to sit with the nucleotides facing up, and then that message from the mRNA is going to be translated into proteins. So the transfer RNA picks up the amino acid that the mRNA is asking for and brings it to it. Now, one way to keep these separate in your head, which has helped me since I learned this in high school and relearned it 50,000 times in college, and every year they just would add more and more detail. How do you remember which one's transcription and which one's translation, right? Because those words are very similar. So transcription is whenever you take your DNA and you convert it into RNA. DNA and RNA are both nucleic acids. They are the exact same, language. They're just like in a different format. So what do I mean by that? So imagine me talking to you right now, or really talking to my screen right now. Imagine that this is the process of transcription. So I'm telling you a message, and you are writing it down on your OneNote so that you can fill in the blanks on your slides, right? So I'm speaking English and you are writing English. One of these is spoken word and one of these is ink on a page, but both of us are writing in English. At least I'm pretty sure both of us are writing in English. If someone else is writing in another language, I applaud you, but this is going to go over your head if you're writing in another language. But I'm speaking English and you are writing English. That is transcription. You're transcribing something from one form to another. Now, in translation, you are taking your RNA, which is a nucleic acid, and you're converting it into a protein, which is made up of amino acids. These are two different languages. So this would be like me telling you something in English and you writing it down in Spanish, or you writing it down in French, or you writing it down in Latin. Whatever it is that you're writing it down in, it's a different language from the one that I'm currently speaking to you. So translation is literally translating from one language to another. Transcription, you're just changing the format. Translation, you're completely changing the language. So hopefully that helps to keep those separate in your head. So I want to talk about transcription first, since it happens first. DNA is going to be transcribed into mRNA. It's important that you use the appropriate language, because if you said translated here, you would be wrong. You have to say transcribed. Again, it occurs in the nucleus. The mRNA will then leave the nucleus and go to the ribosomes. Now, one thing that I have not told you about yet is that the first mRNA that gets made, so from DNA to the mRNA, That first part that gets made is actually called pre-mRNA because we have to do some stuff to it before it's considered like mature, ready to go, ready to leave the nucleus mRNA. And we call it pre-mRNA because every single complementary base is copied, including exons and introns, which again, I haven't told you what that is yet, but don't worry, it's coming on the next slide. What is an exon and what is an intron? But in the pre-mRNA, again, every single solitary complementary base is copied from the DNA into the mRNA. So there are these large sections of DNA that don't code for anything. There's no proteins that are being made. There's no different types of RNAs that are being made. It just doesn't code for anything. And we call those introns. So it does not code for anything. Just a large section of nucleotides that don't have any type of message in them. Okay, those are the introns. The sections that do code for the proteins are called exons. You can remember that the exons are the ones that you keep because exon, the EX could be for expressed. So those are going to be the parts of the DNA that end up being expressed. You end up seeing them at the end as protein. Okay. So those snRNAs that we had talked about earlier, the small nuclear RNAs, their job is to cut out the introns of the mRNA and then put the exons together. And then after the exons get put together, the mRNA will then pick up what we call a cap on its 5' end, so a 5' cap, and it's going to get a tail, which seems silly. It's going to get what we call a poly-A tail. A poly-A tail on its 3' end. What the heck is a poly-A tail? Poly-A means polyadenines. Poly means many, so it's just a lot of adenines in a row. So if we look over here at our picture, we have our original pre-mRNA here. So you can see it has both the exons and the introns in it. The small RNAs, the small nuclear RNAs, are going to come in and create what we call a spliceosome. You don't really have to know that word, but it's super fun to say. But they create this spliceosome, and its job is to cut that intron out. So goodbye, intron. We do not want you anymore. So that intron gets cut out, and the exons are going to be put together. Yay! And then what they're not showing you is that there's going to be a 5' cap over here. It's actually going to be a methyl cap, which is a CH3. You don't need to know that, but if it's a hydrocarbon made up of only one carbon, that's called a methyl group. So it's a 5' methyl cap. And then on the 3' end, they're also not showing you the poly-A tail because they're not really showing you any of the RNA nucleotides. It's just understood that these orange or red, I can't really tell what color, I guess orange. These orange blocks are representing nucleotides. They're just not showing you the letters because that would look very busy. But there's a poly-A tail with a bunch of adenines at the end of this mRNA. So once the mRNA has the introns cut out by the snRNAs and it has its 5' cap and its poly-A tail, that's considered a mature messenger RNA, and it can leave the nucleus. So it's kind of like when, I don't know if any of you guys, if your mom is like this, but my mom was. If I'm trying to go, obviously when I lived at home, but if I was trying to go somewhere and I wanted to wear a certain outfit, I would have to come out and my mom would have to see me before I leave so that she could say that my outfit is a-okay for me to go in public, or she would say absolutely not, go turn around and put some more clothes on. Obviously, as the person I am, I never tried to wear anything that was a little bit too flashy, if you know what I'm saying. I was not a TikTok girl or whatever, with my belly hanging out, but hey, you never know. But it would kind of be like that, like you're mom's not going to let you leave the house until you're dressed how she thinks is appropriate. And so it's kind of like that with the mRNA. The pre-mRNA is not dressed appropriately. It needs to go put on its poly A tail, and it needs to go put on its 5' cap before its mom says, okay, you're good, you can leave. So yeah, if you didn't understand it before, hopefully my very vivid explanation of not showing your midriff in public helped you. So yeah. Okay, so what are the steps of transcription? The first step is to separate the strands of DNA, which is done by our good old pal DNA helicase. After the DNA strands are going to separate, the second step is initiation, which means start. So the initiation occurs when the RNA polymerase binds to a sequence of the DNA, a specific sequence of the DNA actually, called the promoter. It's just a specific sequence of nucleotides that the RNA polymerase recognizes. From there, the mRNA is going to be made in the 5' to 3' direction. That should not be surprising to us. That's the same way replication is done, because the polymerases can only add to the 3' end, because the 3' end is the only one that has a free hydroxyl group to do dehydration synthesis. The fourth step is elongation, so just adding nucleotides, steady, pairing up complementary, A pairs with U, T pairs with A, C pairs with G, G pairs with C. And then the next step is termination, so that's how we end the process. So the RNA will stop being made when the RNA polymerase sees a stop codon. So when it makes the UGA or the UAG or the UAA, once that happens, the RNA polymerase is like, nope, I'm out of here, I'm done, peace out, we're done, this was a stop. And so basically the RNA polymerase just falls off of the DNA, basically, and the DNA winds back up into its double helix, and the mRNA goes get processed, and then the mRNA can leave the nucleus. So you might want to rewind that and maybe listen to it again, or just read the words on the screen again. Don't want to rush you through this, but it's weird because I can't really field questions while I'm talking to myself, so yeah. Okay, so you might be wondering, okay, well, what's the point of having introns and exons in the first place? Like, why would we have introns? What's the point of that if they're just going to be cut out anyway? And this slide kind of explains, well, here's the point. So we have this phenomena called alternative RNA splicing that can occur because of those introns. So if we look at a sequence of DNA, if you look at the top of this picture, you can see exon 1 in blue, exon 2 in green, exon 3 in red, exon 4 in light blue, and exon 5 in orange. And then all of the DNA in between, those are the introns, right? And so then if we look at the messenger RNA that originally gets made, that pre-mRNA, you can see that it has all of the exons as well as all of the introns. And so because of this, we can basically, because of those introns, we can have some proteins that will come in and they can cut out specific introns, and they can sometimes even cut out exons to put specific exons together in order to get different proteins. So in this case, in the first one for protein A, we used every single exon, 1, 2, 3, 4, and 5. In the case of protein B, we only used 1, 2, 4, and 5, so 3 got cut out. In protein C, we used 1, 2, 3, and 5, so exon 4 got cut out. And if you look at these pictures, you can see that all 3 of these proteins look very different from each other, right? So basically what happens is the DNA is going to be transcribed into the mRNA, and the DNA is just going to like be transcribed over and 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 over again. And so basically what you end up having is a bunch of the same mRNA, and then all of those mRNAs can then just be cut down to include whatever specific exons you actually want, instead of having to go through and make one specific mRNA for each and every one of these 3 proteins. you can just use the same mRNA and cut it different ways to get different proteins. So basically, since our cells have to leave the nucleus, this essentially saves us time by making a bunch of these. Whew, just got it in 15 minutes. Go me.